Welcome to A Minder, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Hello, my name is Anusha, and I'll be your host for this episode. We are looking at papers that were published in June of 2020 that fall under the umbrella of disease mechanisms and pathology. More specifically, we're covering 14 papers on metabolism and mitochondrial dysfunction, 4 oxidative stress papers, 2 papers on autophagy, and 2 papers on gut microbiome. We've got a lot of different areas to cover here, but if you find something that interests you, we also have provided a bibliography that is time-stamped for your convenience. So let's get started. Our first topic is metabolism, and we'll be beginning with a paper titled Increased Prolidase Activity in Alzheimer's Dementia, a Case Control Study. The first author is Krishna, and the last author is Subramanian, and this was published in the Asian Journal of Psychiatry. Altered synaptic connectivity has been previously associated with cognitive decline in Alzheimer's disease, or AD, but this study tried to identify prolidase enzyme as a potential contributor to the underlying disease mechanism. Prolidase enzyme catalyzes the final step in collagen metabolism by altering the extracellular matrix, and they found here that prolidase activity was significantly higher in AD subjects compared to the control group. This increase was negatively correlated with cognitive decline, and therefore they suggest that prolidase-induced changes in the extracellular matrix may be an underlying mechanism for cognitive decline in AD. Next, we have a paper titled Fascicle and Glucose-Specific Deterioration in White Matter Energy Supply in Alzheimer's Disease. The first author is Roy, the last author is Desoto, and this was published in the Journal of Alzheimer's Disease. In the gray matter of AD brains, it has been shown that there is reduced glucose metabolism, but glucose and ketone metabolism in the white matter, which is essential for axonal function, has rarely been measured in AD subjects before. The goal of this study was to quantify glucose and ketone metabolism in the white matter of individuals with mild cognitive impairment, AD, or those who served as controls. They did this by using a new approach of combining dual tracer PET imaging with fascicle-specific diffusion MRI. In the context of this paper, fascicles would be bundles of nerve fibers, so white matter tracts. Only the fascicle from the left posterior cingulate segment of the cingulum showed significantly lower glucose uptake in AD compared to controls, whereas ketone uptake was either unchanged or elevated in the brain regions that they looked at. Overall, this study was the first to measure white matter metabolism in AD and found that glucose metabolism was more affected than ketone metabolism. Up next, we have from authors Yu, Wang, and others, published in the journal Advances in Clinical and Experimental Medicine, a paper titled The Role of GLP-1 GIP Receptor Agonists in Alzheimer's Disease. New glucagon-like peptide-1, or GLP-1, analogs have been recently developed and may have neuroprotective benefits for AD. The goal of this study was to show whether the GLP-1 liraglutide could serve as a protective role through tau and BACE1 regulation using both in vivo and in vitro methods of analysis. Liraglutide successfully prevented okadaic acid-induced apoptosis in SHSY5Y and neural cells from AD rats. Importantly, liraglutide protected against okadaic acid-induced cognitive decline, tau activation, and BACE1 expression in AD rats. This study therefore revealed a promising neuroprotective effect of the compound liraglutide in the treatment of AD. Next, we have a paper titled Distinct Utilization of Biotin in and between adipose and brain during aging is associated with a lipogenic shift in Wistar rat brain. The first author is Anand, and the last author is Jayavelu, and this was published in Nutrition Research. Ongoing stress with age has been associated with shifts in metabolic processes. For instance, increased lipid synthesis and storage has been found in the AD brain, whereas adipose limits its capacity for such processes. Contributing mechanisms towards this lipogenic shift from adipose to brain are unclear. Therefore, the current study measured biotin utilization by adipose and brain tissues of young and old Wistar rats. They found that adipose-specific biotin was higher than brain tissue, regardless of age. Interestingly, adipose failed to sustain its biotin stores, while brain tissue increased biotin and lipid content with age. This increase in lipid storage positively correlated with astrocyte use of biotin. The study has potential translational value for changes in metabolic processes when considering age as a risk factor for AD. Next, we have the lipid phosphatase synaptogenin-1 undergoes a significant alteration in expression and solubility and is associated with brain lesions in Alzheimer's disease. The first author is Ando, and the last author is Brian. This was published in Acta Neuropathologica Communications. Synaptogenin-1, or SynJ1, is a lipid phosphatase that plays a role in various processes such as trafficking, recycling, and metabolism. SynJ1 polymorphisms have been suggested to influence the age of AD onset, as well as amyloid-induced toxicity. However, the precise expression and localization of SynJ1 in the AD brain is currently unknown. Ando and colleagues sought to answer this question by examining post-mortem AD brains for SynJ1 protein levels and localization. SynJ1 was mostly accumulated in hyaronobodies, plaque-associated dystrophic neurites, and some neurofibrillary tangles. Interestingly, SynJ1 protein and mRNA levels were higher in AD patients carrying 1 or 2 apolipoprotein E epsilon 4 alleles. From now on, I will refer to apolipoprotein E as ApoE. SynJ1 proteins were also increased in the protein aggregate enriched fraction from AD brains. Remember, you can always refer to the paper for more information, but this study provides data supporting differences in SynJ1 mRNA and protein levels. Additionally, it implicates SynJ1 as a potential therapeutic target and biomarker, especially for AD carriers of ApoE epsilon 4. Next up, a paper titled Polyunsaturated Fatty Acid Deficiency Affects Sulfatides and Other Sulfated Glycans in Lysosomes Through Autophagy-Mediated Degradation. The first author is Wang, the last author is Kamijo, and the journal is Federation of American Societies for Experimental Biology. Sulfatide metabolism and other sulfated glycans are associated with AD. However, the role of polyunsaturated fatty acids, which I'll be referring to as PUFA, which is so much fun to say, you should try it sometime. But to reiterate, the role of PUFA in sulfated lysosomal substrate metabolism is currently unknown. Wang and colleagues determined how PUFA deficiency or supplementation would affect sulfatide metabolism in enriched organs, such as the brain and kidneys in mice. A prolonged PUFA-deficient diet led to a reduction in sulfatides through an increase in sulfatide degradative enzymes in both brain and kidney. This was associated with an increase in autophagy and lysosomal activity. Reduced PUFA intake also led to degraded amyloid precursor proteins, suggesting the activation of the early developmental process of AD. Fortunately, PUFA supplementation rescued the effects of a PUFA-depleted diet. Overall, the study shows that PUFA deficiency increases sulfatide degradation and autophagy in lysosomal function. These findings have important implications for early disease development and potential preventative measures. In the journal Alzheimer's Research and Therapy, we find our next paper published by first and last authors Ashraf and So. It's titled Plasma Transferrin and Hemopexin are associated with altered A-beta uptake and cognitive decline in Alzheimer's disease pathology. Dysregulated heme and iron homeostasis are also a feature of AD. Ashraf and colleagues studied heme and iron homeostasis in 3 groups: healthy controls, MCI, and AD patients. They used non-targeted proteomic analysis by high-resolution mass spectrometry to measure protein levels in plasma samples from the Australian Imaging Biomarker and Lifestyles flagship study of aging, called ABLE, and Kerr Anglican Retirement Village Initiative in Aging Health, or the CARVIA. And so ABLE and CARVIA were the two cohorts that were used. The main finding was that hemoglobin subunit beta was significantly increased in AD compared to controls. Other targets, such as transferrin and hemopexin, were associated with brain amyloid depletion, hippocampal volume, and cognitive performance. Moving on, we have lactate dehydrogenase expression modulates longevity and neurodegeneration in Drosophila melanogaster. The first author is long. They're called long. I don't know how long they are. The last author is Giboltowicz, and the journal is Aging. The journal is called Aging, and I'm sure it's also getting older. I'm sorry, this is just too easy to make these jokes sometimes. Here we're talking about lactate dehydrogenase, or LDH, which catalyzes the conversion of pyruvate to lactate, and it's generally associated with brain energetics and memory formation, so you can assume that it's important for AD. While elevated lactate levels have been reported in aging and AD patients, it's unknown whether this is a protective or maladaptive mechanism. Therefore, Long and colleagues genetically changed LDH levels in adult fly neurons or glia to determine the effects. Increased LDH expression resulted in a significantly reduced lifespan, disrupted locomotor activity, and elevated neurodegeneration. Reduced LDH expression prolonged the lifespan and delayed age-dependent neurodegeneration. The results suggest that LDH potentially influences the onset of neurodegeneration and longevity in flies. So now we're actually changing topics a little bit. So we're moving on from metabolism to mitochondrial dysfunction. The first paper we'll be looking at is by authors Georgievic, Albenzi et al. It's titled Early Onset of Sex-Dependent Mitochondrial Deficits in the Cortex of 3xTG Alzheimer's Mice, and it was published in Cells. The risk for developing AD increases with age, and interestingly enough, the risk is higher in women. Two hallmarks seen in AD are cell loss in the cortex and hippocampus, as well as mitochondrial dysfunction. So the goal of this study was to determine sex and brain region differences in mitochondrial dysfunction in the 3xTG AD mouse model. They reported that changes in mitochondrial function, including decreased coupled and uncoupled respiration, were more prominent in the cortex. Furthermore, these changes were evident at an earlier age in female mice than males. These findings focus on how sex and brain region can influence the translatability of potential therapeutic strategies and disease mechanisms, something to consider. Next up, we have Alzheimer's and Parkinson's brain tissues have reduced expression of genes for mitochondrial DNA OXPHOS proteins, mitobiogenesis regulator PGC1 alpha protein, and mitochondrial RNA stabilizing protein LRP-PRC, also called LRP-130. This is by first author Bennett and last author Keeney, published in Mitochondrion. That would make a really cool band name, don't you think? Here, the researchers capitalized on unbiased screens, which allow for robust generation of potential targets for disease mechanisms and therapeutic strategies. Bennett and colleagues used RNA sequencing in total RNA extracts from the frontal cortical ribbon from AD patients, the ventral midbrain from PD patients, and healthy control subjects. They found a significant decrease in mitochondrial DNA OXPHOS genes in both AD and PD tissues. Further, mitochondria-related targets that were compromised included the master mitochondrial biogenesis regulator PGC1 alpha, complex 1 subunits, and LRP-130, which is a known mitochondrial RNA stabilizing protein. These results implicate reduced mitochondrial ATP production and compromised mitobiogenesis and mitochondrial RNA integrity. Confirmation of the presented targets could therefore lead to disease-modifying therapeutic strategies for both AD and PD. Now we have functional coupling of pre-sequence processing and degradation in human mitochondria. Oh, there's so many umlauts here. The first author is Kukukosi, and the last author is Vogtel. I'm very sorry if I'm pronouncing this wrong. We don't have umlauts where I'm from. Getting back to the science. The mitochondrial proteome is sustained through the import of nuclear-encoded precursor proteins that come with N-terminal pre-sequences. MPP is an essential mitochondrial processing protease that removes the pre-sequences to allow for protein folding and functionality. The matrix pre-sequence peptidase pre-P degrades cleaved pre-sequences and has been associated with AD. This current study characterized the novel HEC293T pre-P knockout cell line to analyze impaired peptide turnover. Pre-P deficiency led to malfunctions in oxidative phosphorylation and altered nuclear expression of stress response marker genes. An accumulation of pre-sequence peptides disrupted downstream mitochondrial processes. Examination of a patient mutation in the pre-P gene that has been associated with neurological disorders showed that protein destabilization makes pre-P vulnerable to severe loss. The results, therefore, suggest that pre-P may be particularly important in human mitochondrial functionality. We've got 2 more papers for this section, the first of which is titled The Distinctive Role of Tau and Amyloid Beta in Mitochondrial Dysfunction Through Alteration in MFN2 and DRP1 mRNA Levels, a comparative study in Drosophila melanogaster. The first author is Naila Kuhlmann, and the last author is Haddadi, and this was published in Gene. 2 hallmarks of AD are A-beta and hyperphosphorylated tau aggregation. However, it's unclear whether the different forms of tau, so there's soluble or hyperphosphorylated, or A-beta differentially contribute to AD disease mechanisms. This study tried to determine which target had the largest effect on the brain antioxidant defense system and genes involved in mitochondrial dynamics in transgenic Drosophila melanogaster. All transgenic flies showed reduced cognitive function and antioxidant activity. So in addition to cognitive function, they also had the paradigm of measuring an eye phenotype, and here they found hyperphosphorylated tau had the greatest effect on eye phenotype, while A-beta had the smallest effect. Additionally, hyperphosphorylated tau significantly altered mitochondrial gene expression when it was expressed across all neurons. Overall, hyperphosphorylated tau appears to exert a more robust toxicity and implies unique disease mechanisms for the various forms of tau and A-beta 42. Now we've arrived at our final paper for this section, which is titled Upregulation of AMPK Ameliorates Alzheimer's Disease like Tau Pathology and Memory Impairment. So this is more in the theme of tau than mitochondria, but it was published by the first author Wang and last author Zhao in the journal Molecular Neurobiology. AMP kinase has already been shown to contribute towards AD pathology. However, its effects on abnormal tau phosphorylation are poorly understood. This study tried to associate AMP kinase levels with tau activity and memory performance using aging C57, 3xTg mice, as well as control mice. They found significant reduction in AMP kinase mRNA and activity in the brains of the aging C57 mice, as well as the 3xTg AD brains compared to controls. Additionally, viral-induced downregulation of AMP kinase in the hippocampus of 3-month-old C57 mice led to increased tau phosphorylation, fear memory impairment, and glycogen synthase kinase 3-beta, or GSK3-beta, activity. When HEC293 cells expressing tau were transfected with AMP kinase or treated with the agonist salicylate, GSK3-beta activity and tau phosphorylation were significantly reduced. Together, this study reveals that AMP kinase levels influence tau activity and memory performance, which could mean that increasing AMP kinase may have a therapeutic benefit for individuals with AD. That brings us to the end of the first half of the papers we'll be covering in this episode. As always, there is a bibliography available with each paper timestamp So if there was a paper you found particularly interesting, you have access to it. Alright folks, I'll catch you on the flip side. We are back, and we now have 4 scrumptious papers on oxidative stress. I will get right into it. The first is titled The Pivotal Role of SUMO1 JNK Tau Axis in an In Vitro Model of Oxidative Stress Counteracted by the Protective Effect of Curcumin. The first author is Bucarello. The last author is Faligione, and this was published in Biochemical Pharmacology. Oxidative stress reflects a toxic imbalance in the cellular redox state and is a contributing factor towards neurodegeneration. Many AD-related processes are impacted by oxidative stress, including protein SUMOylation and JNK and tau protein activation. The authors try to expand upon an interaction between these targets in oxidative stress conditions using SHSY5Y cells. Peroxide induced a dose-dependent increase in cell death and reduced functionality of the cell's degradation system. Oxidative stress also induced aggregation of SUMO1P JNK tau proteins in nuclear bodies. Treatment with curcumin, which is an antioxidant compound found in turmeric, recovered SUMO1, JNK, and tau functions and cell viability. Antioxidant treatment also dispersed the nuclear aggregates. These results implicate the SUMO1 JNK tau axis in oxidative stress and highlights curcumin's potential as a neuroprotective compound in AD. Next, we have another antioxidant, but this time vitamin C. The title of this paper is Vitamin C is a Source of Oxoaldehyde and Glycative Stress in Age-Related Cataract and Neurodegenerative Diseases. The first author is Fan, the last author is Monnier, and this was published in the journal Aging Cell. Oxoaldehyde stress involving methyl glyoxyl production during glycolysis, as well as modification of arginine residues, are 2 major sources of tissue damage in aging-related disorders. A byproduct of these modifications is methyl glyoxyl hydroimidazolone, or MGH1. This study determined whether oxidation of vitamin C, also known as ascorbic acid, was part of this damage. Using liquid chromatography, MGH1 was higher in the hippocampus of AD patients and higher in eye lens and brains of mice expressing the human vitamin C transporter 2. MGH1 was also increased in the substantia nigra of PD patients. Ascorbic acid oxidation was found to be enhanced in AD, which may be due to increased catalytic metal ions in hippocampus during aging. Overall, the authors report enhanced oxoaldehyde stress and ascorbic acid oxidation in aging-related diseases. Alrighty, now we have a paper titled Multiple Herpes Simplex Virus 1, or HSV1, Reactivations Induce Protein Oxidative Damage in Mouse Brain. Novel Mechanisms for Alzheimer's Disease Progression. This is by first and last authors Proto and Palomara, and this was published in Microorganisms. I'm sure the journal Microorganisms is impressive. I would have personally been more impressed by them publishing this paper inside yeast. Herpes simplex virus 1, or HSV1, is characterized by a latent infection that is reactivated periodically. To determine whether repeated HSV1 activations induce oxidative stress in the mouse brain, the current study used biochemical and redox proteomic assays. Markers of oxidative stress, such as 4-hydroxynonenal, or HNE, were increased after repeated HSV1 activations in the mouse model of recurrent HSV1 infections. The authors found that AD-related proteins, such as GRP78 and CRMP2, were disrupted or unable to function following HSV1 reactivations. In conclusion, the results suggest that recurrent HSV1 activation promotes oxidative stress that can lead to neurodegeneration. We have come to our final paper dealing with oxidative stress. The title of this paper is Telomere Length and Oxidative Stress Variations in a Murine Model of Alzheimer's Disease Progression. The first author is Martinez Gonzalez, and the last author is Garcia de la Torre. This was published in the European Journal of Neuroscience. The shortening of telomeres is a hallmark of aging. Since aging is the most common risk factor for AD, the current study wanted to determine whether telomere length was associated with disease severity using an aging transgenic 3xTg AD mouse model. qPCR was used to measure telomere length in brain tissue as well as peripheral blood cells across different ages. Older transgenic mice displayed shorter telomeres in the blood compared to younger mice and wild-type mice. No changes in telomere length were observed in the hippocampus. Oxidative stress increased with age in both transgenic and wild-type mice, but no association was found between oxidative stress and telomere length. The changes in telomere length were worsened in the AD state, but were not impacted by age-related oxidative stress. It should also be noted that measuring telomere length from blood cells may serve as a potential biomarker for AD progression because it's easier to acquire those samples and it's easier to detect this biomarker in the blood. I find it kind of poetic that we ended our section on oxidative stress with a paper on telomeres because telomeres are the ends of chromosomes, are they not? Now let's move on to our two final sections, first of which is autophagy. The first paper we'll be covering is titled Dynamic Changes of Autophagic Flux Induced by A-beta in the Brain of Postmortem Alzheimer's Disease Patients, Animal Models, and Cell Models. The first author is Long, and the last author is he. This was published in the journal Aging. To understand the role of autophagy in the occurrence and development of Alzheimer's disease, the researchers of this paper studied the brains of postmortem AD patients, animal models, and cell models. They found that autophagosome accumulation and lysosomal marker expression were decreased in the brains of AD patients. Using mouse models, they found that in the APP-PS1 double transgenic mice, autophagosomes did not accumulate before the formation of senile plaques, but accumulated along with the deposition of senile plaques. They also observed that the level of lysosomal markers cathepsin B and LAMP1 protein decreased significantly. This result was similar to what they found in the brains of postmortem AD patients. In the APP-PS1 LC3 triple transgenic mice, they found that the number of autophagosomes in the brain increased with age, but autolysosomes did not increase accordingly. In the AD model cells, they found that the fusion of autophagosomes with lysosomes was blocked. Lysosomal functioning was decreased after exogenous A-beta 25 to 35 treatment, as well as with the mutation of the endogenous APP gene. The authors go on to suggest that this may be the main mechanism of autophagy dysregulation in AD. Our second and final paper looking at autophagy is titled NRBF2 is a Rab7 effector required for autophagosome maturation and mediates the association of APP-CTFs with active form of Rab7 for degradation. The first author here is Kai, the last author is Liu. This was published in the journal Autophagy. NRBF2 is a known regulator of components that initiate autophagosomal processes. NRBF2 was localized at autolysosomes and a deficiency of which disrupted autophagosome maturation. Specifically, NRBF2 localized with Rab7 and was responsible for converting Rab7 to its GTP-bound state through its interaction with the Rab7 GEF CCZ1-MON1A. The authors also showed that NRBF2 is implicated in APP C-terminal fragment degradation, as well as amyloid beta peptide production. They suggest that this is done through maintaining the APP and CCZ1-MON1A Rab7 interaction. Overall, the current results identify NRBF2 as a Rab7 effector responsible for autophagosome maturation. This study suggests a novel mechanism implicated in autophagy and AD-related protein degradation. Moving on to our last section, we are now talking about the gut microbiome, and the first paper is titled Elevated Amyloid Doses of Human IAPP and Amyloid Beta by Lipopolysaccharide and Their Mitigation by Carbon Quantum Dots. The first author is Kopel and the last author is Key, and this was published in the journal Nanoscale. In this study, the authors looked at lipopolysaccharide, which I will refer to as LPS, and its catalytic role in amyloidoses. Specifically, they focused on islet amyloid polypeptide, or IAPP, and A-beta, knowing that these two peptides are characteristic of amyloid diseases like type 2 diabetes and AD. They also tried nanomaterial carbon quantum dots as a possible therapeutic agent against these diseases. They did this in an embryonic zebrafish model exposed to amyloid peptides and LPS. They report that treatment with nanomaterial carbon quantum dots rescued impaired embryonic hatching. Perhaps more applicable to Alzheimer's disease, it also suppressed reactive oxygen species production. The improvements are likely due to the binding of these quantum dots to the amyloidogenic and C-terminus region of islet amyloid polypeptide and the N-terminus region of amyloid beta. This is in contrast to the preferential binding of LPS to the N-terminus of IAPP and the hydrophobic core and C-terminus of amyloid beta. We now find ourselves at the final frontier, the final paper that we'll be talking about for this episode, and this lucky paper is titled 27-hydroxycholesterol contributes to cognitive deficits in APP-PS1 transgenic mice through microbiota dysbiosis and intestinal barrier dysfunction. This was published in the Journal of Neuroinflammation by first and last authors Wang and Zhao. The brain-gut microbiota axis is a bidirectional communication highway between the central nervous system and the peripheral intestinal network. There's a growing interest in whether problems in this axis, such as gut microbiota dysbiosis and intestinal barrier dysfunction, would contribute towards AD-related pathology and symptoms. The current paper investigated this possibility using the neurotoxic effects of prolonged 27-hydroxycholesterol, or 27-OHC, treatment in APP-PS1 transgenic as well as in control mice. Microbiome integrity was measured using ultra-performance liquid chromatography, mass spectrometry, and high-junction ultrastructure was examined using transmission electron microscopy. 27-OHC treatment damaged the ileum and colon and compromised the intestinal barrier integrity, which showed a widening of tight junctions and decrease in tight junction proteins. Inflammatory markers were also elevated with 27-OHC treatment. These results demonstrate that 27-OHC treatment compromises the brain-gut microbiota integrity and further implicates this system in AD-related pathophysiology. So there we have it. I'm going to turn it over to Sarah, who is someone who you should be familiar with if you've listened to many episodes of our podcast, which I hope you do. Thank you so much for listening, and it's been a pleasure to host this episode for you. Bye-bye.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMinder. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now member of the AMinder team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, your feedback matters to us. So please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, Thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.